Let's begin with the section 2 of our lecture. And in this second section, we will talk about the histopathology. Histo is the tissue and pathology is the study of any abnormality. So in this section, we will talk about the histopathological changes or the changes in the tissues that can ultimately lead to the carcinoma or cancer of the head and neck. And then also carcinogenesis. Genesis is the formation or development. So how this cancer develop and what are different changes and mutations that occur that can lead to the carcinogenesis of the head and neck cancer. And then also we will talk about some molecular pathology or the changes at the molecular level which can lead to the head and neck cancer. So first when we talk about histopathology or abnormalities of the tissues and then the cells that make up the tissue, the most important thing that tells us the severity of the cancer or the staging of the cancer is whether it is what's the differentiation, whether it's well differentiated, it's moderately differentiated, or it's poorly differentiated. If the cells are well differentiated, you can recognize the cells on microscopic examination, on histopathology, then it means the stage is early and the prognosis of the tumor or the cancer is good and it can be treated or limited to the sites. But if the differentiation is moderate or poorly differentiated, if the cells are not recognized or they are all different types of cells, poorly differentiated, then the prognosis is very poor. So very, very important step in the histopathology is the differentiation of the cells that make up the cancer or the malignancy. So that's the one very important factor. The lymphoepithelioma, this is the tumor. Lymphoepithelioma is developed by the, or it is caused by Epstein-Barr virus, and we already mentioned that Epstein-Barr virus tumors or malignancies, they can affect all age groups. So in young, old, and middle age, all people are affected by this cancer as compared to HPV caused tumor which affect mainly the young people. So lymphoepithelioma is caused by Epstein-Barr virus. Salivary gland tumors, this is the location of in the face close to the ear. This is the salivary gland or the parotid gland, which is the major salivary glands. This is the parotid gland site. So salivary gland tumors can affect the parotid gland, which is the major salivary gland, also affect the minor salivary gland besides the parotid gland. So both major and minor are affected. So this is the location of the parotid gland, which is the major salivary gland. Carcinogenesis, again, this is the parotid gland, parotid duct, and this is the tumor 2 centimeter or less. The salivary gland tumor are usually the benign tumors, so they are more localized as compared to the malignant type of tumor. So this is the parotid gland, and this is the tumor which is less than 2 centimeter, which is 2 centimeter or less than 2 centimeter. The parotid tumors, they are benign. Malignant tumors are adenocarcinomas. So parotid gland involvement leads to benign tumors, but if malignant tumors are the adenocarcinomas. Erythroplakia or leukoplakia is formed. If you see in the previous slide here, this is the lesion that is formed and it is red in color. So red lesion or red ulceration is erythroplakia and white is leukoplakia. Both can occur. Multiple synchronous or metachronous cancer. Multiple synchronous are the cancers or growths that occur at the same time. Metachronous are the cancers that occur at different time. So usually after the treatment, 3 to 4 months, the tumors develop, and that's the metachronous. So different synchronous is occurring at the same time, metachronous occurring at different time. Death from second malignancy can occur, and radiation therapy induced sarcoma. So all these are different carcinomas that can develop, and benign are parotid gland tumors. Then we have malignant are adenocarcinomas. There is erythroplakia, leukoplakia. They can be synchronous, metachronous, and there is radiotherapy induced sarcomas can also develop. Molecular pathology is the abnormalities that can present at the molecular level, very small microscopic level, the abnormalities which takes place at the level of genes as a result of the mutations that can occur at the molecular level. So it can be genetic or epigenetic alterations in the genes that can ultimately lead to the carcinogenesis of the head and neck cancer. There can be overexpression of EGFR, and if there is overexpression of EGFR, then the prognosis of the cancer is poor and the survival is very poor. There is mutation of the p53 that can also lead to the cancer, and also alteration in p13k pathway, which is human papilloma virus positive, which can lead to the alteration of p13, and this can also lead to the development of the cancer. And this HPV positive, and remember HPV associated cancers are mostly occur in the young age and young individual, and HPV is also associated with the cervical cancer in the females, with which the pap smear is performed to diagnose that cervical cancer after the beginning of the sexual life. So this is the molecular pathologies that can ultimately lead to the development of the head and neck cancer. Then also, what are the effects of different molecular changes or mutations that can occur, how they affect? They can lead to mitogenic signaling. Mitogenic signaling is the effect of different chemicals on the growth of the cells, on the differentiation of the cells. So when these genetic and perigenetic mutations are present, they can cause mitogenic signaling, and the response to the different chemicals of the different cells is affected. They can also cause genetic stability, And cellular proliferation, as I just mentioned, the mitogenic signaling can also affect the cellular proliferation, cellular differentiation. So once the cells start getting more and more proliferate and grow more and more, then it's not possible for the cells to get well differentiated with more and more proliferation, because ultimately the cells with the different morphology, different shapes, different sizes are formed. Cells with less viability life are formed, and they are the cells which cannot perform their functions properly, and then they can lead to ultimately not cells which are poorly differentiated, and with the poor differentiation then we have more poor prognosis and mostly lead to the death. So it affects the cellular proliferation and then cellular differentiation, and the cells are produced now which are not fully differentiated. They have no normal shape, size, no cytoplasm, no normal nucleus. Their survival life is very little, and they cannot function properly. So the functioning of the structures and organs are affected. So this was all about section 2. Thank you for watching sqadia.com.